Hello guys, today we'll be talking about botulism and we'll be talking about botulism under the following headings: introduction, causative agents, pathogenesis, epidemiology, risk factors, diagnosis, prevention and control, as well as treatment. What is botulism? Botulism is a rare but potentially fatal neuroparalytic condition that is caused by exposure to the botulinum neurotoxin, which is one of the most potent biological toxins known to mankind. Botulism is characterized by acute, symmetrical, descending flaccid paralysis, which can usually progress rapidly to respiratory failure if left untreated. Botulism is a medical emergency and it is a public health concern due to its high mortality rate when diagnosis and treatment is delayed. The disease is composed of various clinical distinct forms, which include foodborne botulism, wound botulism, infant botulism, iatrogenic botulism, as well as adult intestinal toxemia. Despite its rarity, botulism remains clinically significant because small quantities of the toxin can lead to severe diseases. And also, outbreaks are linked to improper food processing as well as preservation practices. Let's talk about causative agents. Botulism is caused by the botulinum neurotoxin, which is produced by the organism Clostridium botulinum. Botulism is caused by the botulism neurotoxin, which is produced by this organism, Clostridium botulinum. Clostridium botulinum is a gram-positive, obligate anaerobic spore-forming bacilli. It is obligate anaerobic because it has the ability to thrive in environments that lack oxygen. And this is particularly due to the absence of certain enzymes, particularly catalase and superoxide dismutase. And these are enzymes that facilitate in neutralizing ROS, which are reactive oxygen species. And as such, this organism, Clostridium botulinum, and other obligate anaerobic organisms utilize fermentation or anaerobic respiration for energy production. Clostridium botulinum is distributed in nature, particularly in soil, sediment, dusts, as well as in aquatic environments. Other species of Clostridium also produce the botulinum neurotoxin that causes botulism. Some of these species include the Clostridium baratie, as well as the Clostridium botulicum. These are some of the other species of Clostridium that have the capacity to produce the botulinum neurotoxin that is responsible for causing botulism. The Clostridium botulinum there are about 7 immunologically distinct serotypes of the botulinum neurotoxin. 7 immunologically distinct serotypes of the botulinum neurotoxin. We have the type A, type B, type C, type D, type E, type F, as well as type G. Types A, B, E, and F rarely. Type A, B, E, and F cause human botulism. These are the culprits when it comes to causing human botulism. Types A, B, E, and rarely F cause human botulism. Type C and D. Type C and D are involved in animal botulism. They are implicated in most cases of animal botulism. And type G, type G is less commonly implicated in disease. It less commonly causes botulism. Type G is less commonly implicated when it comes to causing botulism. It is important to note that the botulism neurotoxin itself is heat labile. It is a heat labile protein, but the spores of the causative organism, which is the Clostridium botulinum, is highly heat resistant, and as such, it thrives in harsh environmental conditions and in improperly processed foods. Let's talk about the pathogenesis. Let's briefly talk about the pathogenesis of botulism. The pathogenesis of botulism is based on the neurotoxic effects of the botulism neurotoxin, hampering muscle contraction in cholinergic nerve fibers, leading to flaccid paralysis. It is important to note that the route of entry of the botulinum neurotoxin into the body is different depending on the type of botulism. In foodborne botulism, preformed toxins are ingested, particularly in home canned foods, fermented fish, smoked meats, etc. The preformed toxin is ingested, and once this toxin is ingested, the toxin combines with non-toxic accessory proteins, particularly hemagglutinins. Hemagglutinins. The preformed toxins combine with non-toxic accessory protein, particularly hemagglutinin, and this causes the formation of a complex known as the toxin progenitor complex. The progenitor toxin complex, rather, forms a complex known as the progenitor toxin complex. The the preformed toxins ingested in food combines with the hemagglutinins to form a progenitor toxin complex. This progenitor toxin complex helps the toxin to evade the neutralization by stomach acid as well as other digestive enzymes such as pepsin and trypsin. And as such, this toxin gets into the stomach and into the small intestine, and in the small intestine, the toxin gets absorbed into the bloodstream. In infant botulism, in infant botulism, the spores of Clostridium botulinum, the causative agent, enters into the stomach. And because the gut microbiome of infants, particularly infants under the age of 12 months, their gut microbiome is immatured. And so the spores of Clostridium botulinum is able to invade and colonize the gut microbiome, germinating and releasing toxins. These toxins are then absorbed into the bloodstream in the colon or large intestine. In wound botulism, what happens is that the spores of Clostridium botulinum infect the wound, or sometimes due to injection drug use like the black tar heroin, these spores enter into the wound, and then due to the anaerobic nature of the necrotized tissue in the wounds, the spores are able to germinate and release toxins. These toxins are then absorbed through nearby capillaries into the bloodstream. So it is important to note that irrespective of the route of entry, depending on the type of botulism, the end product or the end point is usually the toxin entering into the bloodstream. The toxin, once in the bloodstream, carries out its major pathogenesis, which involves neurotoxic effects on the neuromuscular junction, affecting muscle contraction and leading to flaccid paralysis. I'll use a diagram to explain how that happens. It's also very important to note that once the once the neurotoxin, the botulinum neurotoxin, enters the bloodstream, once the botulinum neurotoxin enters the bloodstream, it is not able to affect the central nervous system. This is because the molecules of the central nervous of the botulism neurotoxin are very large and as such cannot penetrate or pass through the blood-brain barrier. And so they cannot directly affect the central nervous system. But the botulinum neurotoxin affects the peripheral nerves, leading to difficulty or problems in muscle contraction, leading to flaccid paralysis. Let me use this diagram to explain how that occurs, how that happens. This is a nerve. This is a nerve. Okay, this is a nerve. Let's say this is a nerve, a cholinergic nerve. Cholinergic nerve. This is a cholinergic nerve. And then this is a muscle. Muscle. The cholinergic nerve and muscle. Muscle fiber. This is the neuromuscular junction. The neuromuscular junction. The neuromuscular junction. Usually in usually in normal muscle contraction, skeletal muscle contraction, action potentials usually come, and then these action potentials come and stimulate this vesicle in the nerves. The vesicle contains neurotransmitter, particularly the acetylcholine. Acetylcholine. The acetylcholine, once triggered, releases the vesicles rather once triggered releases acetylcholine present in them, and the acetylcholine once released binds to the nerve membrane. This is the nerve membrane. Binds to the nerve membrane. This is the nerve membrane, and the nerve membrane, once it has bounded to the acetylcholine, through exocytosis, transports the acetylcholine to receptors. There are receptors present on muscle fibers. When this acetylcholine binds to the receptors, muscle contraction occurs. But it is important to note that for the acetylcholine to bind to the nerve fibers, it does this through the help of certain proteins known as the snare proteins. It does this through the help of snare proteins. Snare proteins. Examples of these snare proteins include synaptobrevin, synaptobrevin. Also have SNAP25 as well as syntaxin. Binding of acetylcholine to the nerve fiber is facilitated by snare proteins. Some of such snare proteins include synaptobrevin, SNAP25, as well as syntaxin. This is what happens in normal muscle contraction. But when the botulinum toxin affects this process, what happens is that the botulinum neurotoxin prevents acetylcholine from binding to nerve fibers. It does that by inhibiting the snare proteins. So the snare proteins are inhibited in this process. They are inhibited from binding to the acetylcholine is inhibited from binding to nerve membrane by inhibiting snare proteins. This happens through the action of the botulinum neurotoxin. And when this happens, muscle contraction cannot occur. And this is what leads to flaccid paralysis that is observed in cases of botulism. So this is basically the pathogenesis of botulism. Let's talk about epidemiology. Let's talk about epidemiology. Botulism is worldwide, although it is rare, with sporadic cases and occasional outbreaks. Botulism is present in all continents of the world. Its incidence rate is low, but underreporting is also seen in resource-limited settings. Foodborne botulism is associated with home canned foods, with fermented fish, with smoked meat, as well as improperly preserved foods. Infant botulism is the most common in most countries of the world, particularly in the United States of America. Wound botulism is more commonly associated with injection drug use, particularly the black tar urine. Under diagnosis is a problem of in botulism, especially in developing countries, due to limited access to diagnostic facilities, as well as overlapping with other neuromuscular disorders. Let's talk about the risk factors. Certain risk factors predispose an individual to developing or acquiring botulism. Some of such risk factors include improper home canning of foods, also consumption of preserved foods without adequate heating, very important. Also, poor food hygiene practices is also a predisposing factor, and infants under the age of 12 months. This particularly predisposes these infants to infection and evasion and colonization by spores of Clostridium botulinum because their gut microbiome is not properly matured yet. Also, being immunocompromised status predisposes to botulism, and use of unregulated botulinum toxin for cosmetic as well as therapeutic purposes are all risk factors that predispose to acquisition of botulism. Let's talk about the diagnosis of botulism. Botulism is primarily diagnosed clinically, but it is also supported by laboratory confirmation. Clinical features of early botulism include diplopia, diplopia, include diplopia. Diplopia is a is an ophthalmological condition that causes a single object to be seen double. That's what diplopia is, and it is a clinical feature of botulism. Also, blood vision, as well as ptosis. Ptosis is also a clinical feature of botulism, and ptosis involves the drooping or sagging of bodily organs, particularly the breast and eyelids. So arthria, this arthria is involves difficulty in articulation of words due to disturbance of the structures that are responsible for modulating voice into speech. There's also dysphagia. Dysphagia. Dysphagia involves difficulty in swallowing. These are basic clinical signs of early botulism. Also, in severe cases, there is flaccid paralysis and respiratory muscle involvement. Laboratory diagnosis of botulism involves detection of the botulinum toxin in serum as well as in stool, in gastric aspirates, and also in food samples. The MBA, the MBA, that is the mouse bioassay, is the gold standard in the laboratory diagnosis of botulism, but it is actually ethically limited. It involves the inoculation of sample into mice to evaluate the signs and symptoms of botulism after about 4 days of incubation. It is actually ethically limited. Also, ELISA and PCR are also involved. I also use as diagnostic methods. ELISA, which is the enzyme-linked immunosorbent assay, detects antibodies that are specific for the botulinum neurotoxin. PCR, which is the polymerase chain reaction, detects genes that are responsible for coding the production of the botulinum neurotoxin. Also, culturing is another important diagnostic approach. Culturing is done using Culture is done using samples like stool as well as wound swab, and they are cultured. These samples are cultured on selective media, particularly the CBI agar, which is the Clostridium botulinum isolation agar, or its modified form, which is the the BSM, which is the botulism selective medium. These are the medium that are used, the media that are used, selective media used for isolation and detection of the botulinum, of the Clostridium botulinum bacteria. Now let's talk about prevention control. Prevention of and control of botulism is principally based on proper food processing and canning techniques. Also, adequate heating of preserved food, usually to about 85 degrees in about 5 minutes. And also proper employing of food hygiene practices and principles. Refrigeration of perishable foods as well is also very impactful and helpful in controlling botulism. Infants that are less than 12 months of age should not be given honey or honey containing products. This helps to prevent infant botulism. It is also very important to note that prompt cleaning and treatment of wounds is also very pivotal. This helps prevent wound botulism. Let's talk about treatment. Botulism is a medical emergency. It is a medical emergency and it requires urgent hospitalization, usually in the intensive care unit. Very important. In severe cases, supportive care is employed, particularly mechanical ventilation, as well as nutrition and fluid support. Antitoxin is used in terms of managing adult botulism. Antitoxin and the human-derived BIG, that is the human-derived botulism immune globulin, is used in managing infant botulism. It is important to note that these antitoxins neutralize the circulating toxin in the bloodstream, but they do not reverse already established paralysis. Very important to note. Antibiotic therapy is used in wound botulism and not in food botulism or infant botulism. The antibiotic of choice penicillin, is penicillin as well as metronidazole. The antibiotic of choice for wound botulism is penicillin and metronidazole. It's important to note that antibiotic therapy is only used for wound botulism. This is to kill the spores of the Clostridium botulinum that is present in wounds, in necrotized wounds, and that are multiplying, producing toxins. Antibiotics are not used in foodborne botulism and infant botulism because this causes death of the bacteria within the intestine, within the guts, and this leads to more production of toxins that can further lead to severe paralysis. So this is why antibiotics are not used in foodborne as well as infant botulism, but they are used in wound botulism.